Hello, everyone. Thank you for joining us today as we kick off National Tick Awareness Month 2021. Many of you may remember in 2020, our theme was Tick Season Can Be Every Season, with a focus on when ticks are active in our country. In 2021, we are taking the tick discussion from when to where, focusing on tick geography and habitats and encouraging us all to say, could ticks be there? Be tick aware. My name is Nicole, and on behalf of the Canadian Veterinary Medical Association and Merck Animal Health, I would like to thank you for joining us today as Dr. Katie Clow shares her results from the Canadian Pet Tick Survey and provides information on the development of an evidence-based approach to ticks and tick-borne disease in your practice. In support of our webinar today, I would like to read a few words from the Canadian Veterinary Medical Association. The CVMA is proud to continue to work in partnership with Merck Animal Health on the National Tick Awareness Month campaign for the 6th year. Expanding tick populations and territories make it more important than ever to arm veterinary professionals with resources to help clients protect their pets, and in turn their families, from the diseases ticks transmit, including Lyme disease. We look forward to cultivating this partnership and enhancing these tools for years to come. Before we get started with today's presentation, there's a few housekeeping points to share. First, as a reminder, today's presentation has been approved for 1 hour of CE credit. Following the presentation, there will be a short quiz which you must complete to receive your CE certificate. Throughout the presentation, you will be able to ask questions by pressing the question mark icon on your screen and typing in your question. Dr. Clow will respond to your question at the end of the presentation if time permits. Finally, this webinar will be archived if you would like to revisit portions of the presentation or have staff that were unable to attend today. In support of National Tick Awareness Month 2021, all clinics across Canada have received an email communication regarding the tools available to promote tick awareness in your clinic. These tools can also be accessed on the Resources tab on your webinar screen today or through the CVMA website. The awareness tools this year include a tick activity calendar, which you can personalize to your clinic location, highlighting the potential days in the last 12 months where ticks may have been active in your area. A shareable animated video highlighting the 2021 campaign theme and encouraging pet owners to be aware of where ticks may be found. And we've also created a social media toolkit with ready-to-share social media posts for the most popular platforms. In addition, we invite you to visit the updated www.ticktalkcanada.com website, which provides valuable tick education materials and videos for your pet owners, including an interactive regional risk map, where you can select the province or region and be provided with relevant tick information. We hope you find these new tools helpful to support your clinic tick education activities in 2021. For today's webinar, we are fortunate to have an expert in her field to speak with us about tick risks in Canada and the important role we play in the veterinary profession. Dr. Katie Clow is a graduate of the Ontario Veterinary College and currently holds the position as Assistant Professor in the Department of Population Medicine at the college. Her research focuses on the ecology and epidemiology of zoonotic and vector-borne diseases. She is passionate about One Health and eco-health and strives to make a positive difference in the health of animals, humans, and the environment. And now I will pass it over to Dr. Clow to get started with today's webinar.
Great, thank you. Everyone can hear me okay? Okay, so today, as Dr. Coppolino was just mentioning, along with the theme of knowing where ticks are, the title of my presentation today is Location, 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 looking at regional patterns of tick and tick-borne pathogens in Canada. And so happy National Tick Awareness Month, and thank you for the opportunity for me to come and speak with you and share the findings from some of the recent research that's occurred in Canada. So our roadmap for the next hour, first I'm going to give you an overview of the basics of the Canadian Pet Tick Survey, which is the recent study conducted by our lab, and then focus specifically on regional patterns of key genuses of ticks Ixodes, dermacenter, and then a couple of our invasive species, the amblyomma and the rhipicephalus. Then I'll highlight some additional findings, place this in the context of also national active tick surveillance, and then think about how to apply these to practice. And hopefully there'll be some time for questions, as long as I don't ramble too much. So as was mentioned, the objective today is to be able to take all this information and use it to develop an evidence-based approach for ticks and tick-borne diseases in your practice. So as we jump in, I want to give you just a brief overview of the Canadian Pet Tick Survey. So this was a study that was conducted out of the Ontario Veterinary College. Myself and Dr. Scott Weiss were the leads on this study. And this was a year-long study that collected ticks from pet owners through participating veterinary clinics across Canada. And it was generously funded by OBC Pet Trust, and really only possible because of the great response that we got from veterinary clinics and clients. And so the rationale for conducting this national survey is really, you know, it's not a surprise. We all know that there have been rapid and ongoing changes in tick abundance, distribution, and pathogen infection prevalence. And this is really keeping us on our toes, and we need ongoing collection of data to be able to understand what the risks are and be able to incorporate that into our veterinary practice. And so the key objectives for the Canadian Pet Tick Survey were to establish a baseline of the distribution of ticks on Canadian dogs and cats across space and time, to assess the patterns of tick species infestation on dogs and cats in Canada, and determine the pathogen risks from ticks on cats and dogs in Canada. And as you can imagine, there was a lot of data that was collected in this study. Today, we're mostly going to focus on objectives 1 and 3. Objective 2 is still very much in the data analysis stage, but we can highlight some basic findings. And again, there's going to continue to be data that's generated from this study. And so our approach was, we went out and we recruited clinics from across Canada. We were aiming for about 90 clinics across Canada with specific geographic targets. The response was overwhelming, which was great. And unfortunately, in some regions, we had to turn some people away. And based on varying responses, some regions had more representation than others, just based on what clinics wanted to participate. Fortunately, we had clinics in every province across Canada and lots of different regions, so we had a nice geographic scope. And how this study was conducted was we sent every participating veterinary clinic a kit for collection, and they would mail back ticks with accompanying just a short survey from the owner in batches every month, or more frequently if there was a lot of ticks. And so on the left-hand side, you see the envelopes that were frequently filling our fridge at the Center for Public Health and Zoonosis at the University of Guelph. And then all of these samples, they were unpacked, organized, morphologically ID'd using a stereoscope. And then if they were part of the study that would be if they were tick species that we were interested in testing, they would then go back to the lab, get made up into this tick slurry that you could see on the right-hand side, and undergo PCR pathogen detection. So we had a great response. We had over 2,800 submissions. And so one submission means one pet, but it could mean multiple ticks. So we get one survey per pet, and it could be anywhere from 1 to 50 or more ticks that were on that animal. And we, in total, collected over 4,500 ticks. So as you can imagine, there are a lot of weekends spent at the stereoscope looking at all of these great samples that were submitted. And really, I'd just like to give a shout out at this point to the fact that this study has really taken a team, and the team has continued to grow. When the study was sort of just getting rolling, we had 2 undergraduate students that were working in the lab with me, and were worked off their feet unpacking envelopes and sorting samples and keeping up with some of the testing. And then two of these students moved on to do their undergraduate theses with me in the fall and winter. And so this was really great research experience for them. And they were really instrumental, again, in processing a lot of these samples. As you can imagine, COVID-19 has had its impact on this study. Fortunately, the study ran from April 1st, 2019 to March 31st, 2020. So it was only the last month of the study that was really impacted, but our ability to be in the lab and process samples and work with the data was severely limited from March moving forward into the summer. We had one student veterinarian who was working with us, collating some of this data and working on infographic production, which I'm going to talk about at the end of the presentation. But we were really delayed in our ability to get into the lab until the fall. And so I now have two master students that are working with me on this study, which is really exciting. But there's just some data that has taken us a while to process and get ready because of these delays from COVID-19, which I'm sure you all understand. And before we jump into the data, I want to talk a little bit about the potential limitations of this type of study. So it's great that we have all of this data, but we are still restricted in our geographic distribution. We can't collect from everywhere across Canada. Of course, that would be great if we could. And so there are certain regions that we are targeted in that have stronger representation than others. And so this needs to be considered in light of the findings. The submission timeline was only for 1 year, and so although it does provide us some nice seasonal data and fluctuations in seasonal patterns, it's not longitudinal data, and we know that even on an annual basis from year to year that our tick populations change, and so this is a snapshot in time essentially. And finally, this is essentially passive surveillance. So individuals that find ticks on their pets send them to the veterinary clinic, and the veterinarians submit them for us. And this has a lot of benefits. It allowed us to collect a lot of samples in a relatively short period of time. It allowed us to engage with pet owners, and that's a really great outcome of it. But again, there is some response bias that's introduced. People are going to submit if they're interested and aware of the study, and so that's going to vary by region. There might not be full details that are included on some of the survey responses because people just don't know the answers or they forget. And again, the population is going to vary by region as well. And we generally have more favorable responses to passive surveillance in areas where the population is higher because there's just more people to submit. So those are all things that can sort of impact our data collection and need to be considered as we're looking through our results. So now I'm going to jump into our regional patterns, looking at distribution, seasonality, and infection prevalence for some of the most common findings that we had. And so before we jump in, I just want to review a little bit about regions. So we tried to classify the data to as fine a scale as we could based on the number of samples that we had. And so for many provinces, we didn't have enough samples or enough clinics submitting that really we could go finer than the provincial data. So for British Columbia, we've classified mainland and Vancouver Island as two separate areas and have data for both. For Alberta, Saskatchewan, and Manitoba, it's strictly at the provincial level. In Ontario, because we did have a large number of clinics and there's a big population in Ontario, we have the Northern Ontario region, the Eastern Ontario region, the Central Ontario region, the GTA, and then Southern Ontario. And so I would just want to make note of where those are, and these are sort of classified just based on grouping clinics together, not based on sort of defined geographic borders. And then Quebec is predominantly Southern Quebec, but all lumped together, and same with our maritime provinces, it's each on the provincial level. Okay, so for our Ixodes genus of ticks, the main samples that we got were black-legged ticks, western black-legged ticks, and groundhog ticks. And I'm going to show you a little bit where some of those key findings were. So To present these, I've put together a series of charts. How to read these charts, it's important to sort of take in two parts of the data together. So the first column tells the total number of ticks submitted and ID'd, meaning there were some ticks that were submitted that, they just weren't in good enough quality for us to actually ID them. Those numbers are few, but that captures the ones that we actually had solid ID on. And so as you can see here for Western Canada, we received a lot of submissions from mainland British Columbia, a smaller number from Vancouver Island, a ton from Saskatchewan, and then just a handful from Alberta. So that's important, and we need to consider two things there: the actual tick population and a high abundance of ticks leading to more submissions, and the other things that I discussed, the fact that we do have, it's based on interest and whether people are looking and thinking about ticks, whether they're going to submit. So both of those play into the numbers. And then the proportions are essentially the percentages of those total numbers. And so as you can see in British Columbia, a good proportion of those were Ixodes pacificus, or the Western black-legged tick. In Vancouver Island, all of the submissions were Western black-legged ticks, so clearly a higher area, favorable habitat and exposure on Vancouver Island. But about a quarter of the submissions from mainland British Columbia still came as Western black-legged ticks. If we look at Alberta, we have 7%, so it's essentially one Ixodes pacificus and one Ixodes scapularis were submitted. Both of these had travel history and were not locally acquired from Alberta. And then in mainland British Columbia, the other sort of Ixodes species that we're seeing were predominantly Ixodes angustus. They're not a super common species, but they're enough to pay attention to because we know that Ixodes angustus does have the capacity to transmit Borrelia burgdorferi, which is the causative agent for Lyme disease. And so even though they were much lower in the percentage, we still pay attention to them because of their vector potential. And in Alberta, the other sort of large number of other Ixodes species were predominantly Ixodes marmotae, which are a species that we generally see on wildlife and were found on dogs that were sort of more roaming outside. And as you can see in Saskatchewan, we only received one Ixodes scapularis. The rest were a different species. And so we can see from that chart that we have a significant a potential significant risk of Western black-legged ticks predominantly in British Columbia and much less risk in Alberta and Saskatchewan. If we move into central Canada, again, this chart's organized the same way. We have Manitoba on the top, Quebec on the bottom, and then our regions in Ontario in the middle. And you'll see that the numbers have switched a lot. And so in most regions in Ontario, we have more than 50%, upwards of 70 to 80% of all the ticks that were submitted were Ixodes scapularis. And so clearly a prime risk for dogs and cats in Ontario. In Manitoba, we still received quite a few Ixodes scapularis, less percentage, but we did receive almost 500 ticks from Manitoba. And then in Quebec, a large percentage being Ixodes scapularis. And so the clinics that we did have submitting were predominantly from southern Quebec, and so Ixodes scapularis being a large risk here. If we look at the total numbers, Eastern Ontario by far wins the total submission. We know that Eastern Ontario is an area of high black-legged tick risk and growing and expanding. And so our study continued to illustrate that Ixodes scapularis in this region is definitely an issue. And same in Quebec, if we look at the number of submissions we received from Quebec. In Quebec, the other Ixodes species of interest that shouldn't really be left out of the conversation is the groundhog tick. And so these are predominantly nest-dwelling ticks that we don't think of a lot actually as pests for companion animals, but we did receive quite a few of them from companion animals in Quebec. And now if we go into Eastern Canada, we have quite similar findings that we saw in the central part of Canada, where a large proportion of the ticks that we received were Ixodes scapularis. If we look at sort of bulk numbers, Nova Scotia, again, for a very small geographic province, we received a lot of ticks, almost 700, and close to 40% of these ticks were Ixodes scapularis, and we know that this is a highly endemic region for Ixodes scapularis, and so a risk for cats and dogs in Nova Scotia. Prince Edward Island, although we didn't receive a huge number of ticks, the majority of those were Ixodes scapularis, and I'm going to share some data as we move along that Prince Edward Island may be a sort of emerging area for black-legged tick risk, and that this is being backed up by field surveillance as well. And then in New Brunswick, almost all of the ticks submitted were either Ixodes scapularis or Ixodes cookii. So those were the predominant tick species that we're seeing submitted from New Brunswick. In Newfoundland, I should say Newfoundland and Labrador, my apologies, we received one submission from Labrador, which was Ixodes scapularis, but this dog had a travel history, and so none of them were locally acquired. Sorry, I'm talking through without using my animations. In Prince Edward Island, the other Ixodes species that made up about 10% of the submissions were Ixodes murus and Ixodes marxi, which are predominantly wildlife feeding ticks, but we do occasionally see them on dogs and cats. So not a surprising finding, but something to make mention of. Okay, so now as we've talked about these tick species, I'm going to bring you our laboratory findings, and I want to sort of prepare you on how to read these maps. And so, sorry, I think there's an animation here, it might click ahead, but in these maps, there are circles that demonstrate the size of the submissions that we got. So the bigger the circle, the more ticks that we received. And then the colors illustrate from light to dark the increasing infection prevalence. And why that's important is, you know, if we received only a handful of ticks and tested them, and let's say we had 4 ticks and 2 were positive, that gives a really skewed result that the infection prevalence is 50%. And maybe that's what we found, but we have no confidence in that estimate. And it's, you know, it can change as we increase our sample size. And in the tick world, generally we want to see about 100 samples before we have a reliable infection prevalence. And so in some regions that just wasn't possible. We didn't receive that. And so I think it's important that as you look at these maps, you consider both the size of the circle and the color of the circle together so that you get that confidence in the estimate as well. And the other thing I should note is that some of our ticks were pooled when we tested them. And so it was, if they were pulled off of the same animal and they were the same life stage and same sex and same species of tick, then they would be pooled together. And so to calculate our infection prevalence, we used a special pooled inflation rate that's developed by a statistician at the Centers for Disease Control and Prevention. Sorry, I clicked back. Okay, so here's sort of our overall map. And if we click into our western provinces, we see most of our samples from Saskatchewan and Alberta. You know, there weren't very many samples, and so none of those were positive. Sorry, I think there's some animations here that will click through, unfortunately. And then in British Columbia, we see it about at 11%. And so a little bit higher than typically seen, but still quite low. And then in Manitoba, our overall was about 22%. If we move ahead into Ontario, we see differences based on region. The overall was about 23%. And then in Southern Quebec, it was about 25%. Sorry. I don't know why this keeps doing this. But we see quite a lot of differences in Ontario, where Eastern Ontario, we have a lot more ticks, so we're more confident in our infection prevalence. And most of these were quite high infection prevalence, so 20 to 30% and greater. But if we look into Southern Ontario, we're more around, the 10 to 20%. In the eastern provinces, again, we see sort of a similar trend. PEI was a lot lower. In New Brunswick, I'm not ready, sorry. In New Brunswick, we had about 22.6% were positive. In Nova Scotia, about 20%, and then in PEI, it was lower at 11%. And I'll put that into context in a second. If we flip ahead to our Anaplasma distribution, the interpretation of these maps is the same with the circle size and the color. In the West, we see very low, about less than 1%. And then in Manitoba, it's a little bit higher, particularly in that southern area, where it's up around 7 or 8% of infection prevalence for Anaplasma. In Ontario, we see Southern Ontario being very, very low, down under 1%. And then in Eastern Ontario, we see a little bit of hot spot for Anaplasma, where it's starting to go into that higher percentage. And then out east, we see in Nova Scotia and New Brunswick, again, still that quite low, less than 5%, but PEI was actually quite a lot higher, at around 14%. And so as I ramble off numbers, sorry about the animations in my slide, I didn't realize they were in there. What do those numbers actually mean? And so when we think about Borrelia burgdorferi, in most cases, once it gets up over 20 to 30%, we're really thinking there's an established transmission cycle between the black-legged tick and wildlife reservoir species, and that risk is quite strong. So that's, like 1 in 4, 1 in 3 ticks is carrying it. And at that time, we, decide that it's established there, it's a high risk, and we really should be thinking about it. Lower, it could be emerging, and we also have sort of adventitious ticks that can be added into the population from, bird-borne ticks or wildlife ticks. And so that's certainly an area to monitor. For Anaplasma, we see infection prevalence a lot lower than for Borrelia. And it seems to be sort of an emerging pathogen where we're starting to see it increase in certain areas of Canada. And so the areas that I had mentioned, being Southern Manitoba and Eastern Ontario, are two of those sort of prime regions where we're seeing the infection prevalence change from, hovering around 1%, going up into the 5s and the 10%. So something to keep aware of. And certainly as you're thinking about tick-borne disease, adding that differential to your list is really important. In Prince Edward Island, the finding of that high infection prevalence, we don't have as many ticks that we tested, so the confidence of our estimate needs to be taken into consideration there. But we also need to do some more field surveillance to understand if this is an emerging risk in that area and that we're consistently finding Anaplasma in ticks in Prince Edward Island. And so that requires additional follow-up, but it's a really good starting point for further research. Okay, so now I'm going to move into our next genus of interest, which is Dermacenter species. And in Canada, we predominantly think of 3 different tick species. So that's the Rocky Mountain wood tick, or Dermacenter andersoni, the American dog tick, or Dermacenter variabilis, and then the moose tick, or the winter tick, which is Dermacenter albopictus. The challenging thing for these guys, particularly for someone in my field, is that they look exactly the same, except you can differentiate them based on the size of the goblet cells in their spiracles, which if that just sounded like gibberish, that's okay. And so they have these anatomical features that are hidden under their back leg, but even that is a bit subjective. And previously, we used to really, we'd see a tick like this and think about the geography where the tick is being submitted from and use that a lot to help guide our identification. And the results of our study show us that we might want to be a little bit more hesitant about just using geography to identify these ticks. So if I moving ahead. There we go. I think I pressed the wrong button. So in Western Canada, so British Columbia, this is the mainland because remember Vancouver Island, we only had Ixodes pacificus submitted. Over 60% were Dermacentor species. And previously we would have thought that all of these were Dermacentor andersoni, or the Rocky Mounted wood tick. That's typically the only thing that we found in British Columbia. But we conducted species-specific PCR to determine, to sort of back up our morphological ID, because as we were looking at these ticks, we're fairly confident that they weren't just Dermacentor andersoni. And lo and behold, almost 75% of these ticks were actually Dermacentor variabilis, or the American dog tick, so very uncharacteristic to what we typically see. And there's been a recent study that was conducted in the States where similar sort of range expansion of Dermacentor variabilis and sort of taking over where Dermacentor andersoni have been found has also been noted. And so this is a really great starting point for additional research. We don't know if it's sort of Dermacentor variabilis moving westward in Canada or Dermacentor variabilis moving northward from areas of the States, or maybe a combination of both. You know, we have some pretty big mountain ranges in there that are, you know, that interfere with dispersal, but we need to do more research, do some more genetic analysis on these ticks to understand this process. It's really interesting though, and one of my master's students is actually going to be investigating the findings of this research and conducting more research on this phenomenon further. In Alberta, we got a mix of Dermacentor species, and so we're again seeing sort of that range overlap of both the Rocky Mounted wood tick and the American dog tick. And then in Saskatchewan, we're only getting the American dog tick, or Dermacentor variabilis. So again, we're sort of seeing Dermacentor variabilis take over the range of the Rocky Mounted wood tick, and we're not really seeing expansion of the Rocky Mounted wood tick. In Eastern Canada, all the Dermacentor species were Dermacentor variabilis, and we see a lot of these guys being submitted from southern Manitoba. The majority of the ticks there, with the rest being Ixodes scapularis, as I had mentioned. In southern Ontario, we're seeing, you know, about a third of our submissions being Dermacentor variabilis. Nova Scotia, a lot, very active Dermacentor variabilis population in that year, involving, you know, almost over 60% of our submissions. And then very few being submitted from the provinces that are a little bit more northern. So southern Quebec, New Brunswick, and Prince Edward Island, very few submissions of our Dermacentor variabilis. The other species that I did want to touch on, because I think the results of our study are encouraging, but also encourage us to remain vigilant. So we did receive a number of species of sort of our invasive tick species, the ones that we don't think are established yet in Canada. And all of these submissions had a history of travel. So that's really valuable. We didn't find, you know, patterns where multiple animals from the same region had multiple submissions of these ticks from them, which would make us want to go out and do field surveillance and see if potentially there was a population there. And so the Lone Star tick is sort of the biggest one that we would be concerned of, because we do know, particularly sort of in southern Ontario and southern Quebec, that the climate is conducive to these guys surviving, and they do get introduced by wildlife and bird species, and so we need to remain vigilant for them. But right now, our study didn't show any evidence that there are potentially reproducing populations of these guys. We didn't receive any submissions of the Asian longhorn tick, which is great. This is the tick that's sort of rapidly spreading across the United States. Reproduces on its own and is a little bit of a scary pest for particularly livestock species. And so that's, again, another positive finding. The most common invasive tick species that we received was the brown dog tick. And I think this is a really important finding for us to know that we need to remain vigilant. You know, we're not particularly concerned that this guy is going to set up shop outside in many areas. It's mostly found in tropical areas. But the thing is that if you know anything about the brown dog tick, this guy can happily survive inside and will create in-home infestations. And so all of these, there's a large portion of dogs that were imported from different areas, either from the States or from tropical regions that had these ticks on them. And the sort of scary thing about these things, if they're not noticed really early, a lot of times if dogs come in, they have heavy infestations and there's already a problem in the home. So this is a good discussion that if you know that someone is bringing in a dog from a tropical area, that these guys are a risk, they should be on tick prevention that specifically addresses this type of tick. And it's a really important for the owners to remain vigilant in their homes as well. Because I don't think anybody really makes a furry addition to their family and hopes that they get an infestation in their home at the same time. So that's never a particularly good thing. I'm going to jump into some of the seasonal patterns just to reinforce what we talked about last year and last tick awareness month. And so in Western Canada, we see a typical really heavy tick burden in the spring months. And so from April, even starting sort of in March, up until July, August, we see a lot of submissions. And this is really driven by the phenology or that seasonal life cycle fluctuations of the two main ticks we have out there, being the Western black-legged tick and the dermacenter ticks, which are, these guys are most active in the spring. And so, not saying that there's no other activity, we still received some submissions into the fall, but the bulk of that activity is really driven in the spring. In central Canada, and we'll see the same thing in eastern Canada, we have this bimodal activity. And this is really driven by both dermacenter activity, Dermacenter variabilis. The adults are almost exclusively seen in the spring, and then black-legged tick activity, where black-legged tick adults and nymphs are predominantly active in the spring, and then the adults are active in the fall. And so that really drives the two patterns we see where, March, April, we start to see an increase into May and June. By July, August, September, these are our hot, dry summer months, not particularly great for these species of ticks. And so we see a die-off in activity. And then come October, November, we see that peak again. And so if you look at the Quebec numbers, we see a little bit more activity in the summer months. And if you recall, I talked about Quebec and New Brunswick, there being a lot of groundhog ticks as well. And groundhog ticks are particularly active spring and summer, and then they just sort of go into a quiet period. And so some of this summer activity is also driven by the groundhog tick. And then if we move into eastern Canada, again, we see that bimodal spring, fall. And again, where we look at New Brunswick, there's a little bit more activity in the summer months because we had more Ixodes cookii, or the groundhog tick. Okay, and now I'm just going to jump into a couple other key findings that we had on tick infestation patterns. The first being, let's not forget about cats. And so surprisingly, of all those submissions, 16% of submissions were from cats. And so it's not just a handful, it was a significant number of cats presenting to clinics with ticks on them, or owners at home finding ticks on their cats. And so this is just a really important finding to continue the conversation about paying attention for risks of ticks on cats. A lot of people have heard the myth that cats just groom ticks off, and clearly this isn't the case. We did see variation based on region, and so in Eastern Ontario and Quebec, New Brunswick and Nova Scotia, more than 20% of our submissions were from cats. And actually, I think New Brunswick was the highest with over a third. and smaller amounts in mainland British Columbia, certain parts of Ontario, and then in Saskatchewan. And particularly in Saskatchewan, where we're really seeing only Dermacentor variabilis submitted. Those guys just love dogs. And so that's not a particularly surprising finding. But it doesn't mean that cats then fall off the table for the conversation. The other finding that we had was if we looked at location of tick acquisition, so on our survey sheet, owners marked or veterinarians, whoever removed the tick, on their sheet where the tick was found. And so of all our submissions, more than 50% were found on the head and neck. And this is a really important finding. You know, if we're talking to owners about the importance of doing a tick check, and, you know, people have limited times in their day and they want to get the biggest bang for their buck, look at the head and neck, check inside the ears. You know, dogs and cats love to go rooting around in things that are generally tick habitat. And so if, you know, you only have time after your walk before you run off to work to look there, that will give you at least some assurance that you're getting the bulk of the ticks, and hopefully you have time for some additional tick checks later on in the day. Okay, so that sort of wraps up my discussion of the Canadian Pet Tick Survey. And I wanted to put this in context with some other additional surveillance that's happening for ticks on a national level. And so the Canadian Lyme Disease Research Network is a national network of researchers that are interested and study Lyme disease. And it's quite a transdisciplinary network with, you know, physicians, veterinarians, epidemiologists, entomologists, people in labs, patients, frontline physicians, you know, a large group of individuals that are really involved with Lyme disease. And so part of this network has a key component in surveillance. And so the Canadian Lyme Sentinel Network was established in 2019, and there's at least one Sentinel region in each province where they're collecting data on ticks and Lyme disease. As you can imagine, no field work or very limited field work was done last year because of the shutdowns due to COVID-19. But there's some interesting findings to share from 2019 and can be considered in light of our findings in the Canadian Pet Tick Survey. And so I just shared that this publication just recently came out. As you can see from the long list of authors, it was a highly collaborative process. This is through the CCDR, which is open access online. And so you can just Google this and it will have access to all the maps and all the data. And so this map shows the Sentinel regions. And as I said, there is at least one Sentinel region in each province. Ontario has a few more because Ontario has a high population. And the circles demonstrate the size of the circle, shows how many black-legged ticks were found in that area, and the color coding is just based on life stage. And so as you can see in sort of mainland British Columbia, we're finding a decent number of ticks, and then in Eastern Ontario and into Nova Scotia, New Brunswick, Southern Quebec, a very large number of black-legged ticks being detected by active surveillance. So this is going out and tick dragging in certain locations. If we look at our infection prevalence, there's some interesting findings and help us interpret our study as well and know, you know, where we need to focus more research efforts. And so in Vancouver, no Borrelia or Anaplasma detected based on a small number of samples, but consistent with years and years of surveillance that the infection prevalence is quite low in that area. Again, no black-legged ticks detected in the Sentinel regions in Alberta and Saskatchewan, and only a small number actually collected around Winnipeg. And none collected around Hamilton, Ontario, or just 2 collected and none found. So that's an area where we've been doing work. It's right around Guelph, and we're seeing some changes. So it'll be interesting to go back out this year, which is the plan, and collect more data. Then we see sort of our Eastern Ontario, Southern Quebec spots where our infection prevalence is very high, 30, 40, 50%. And then same high findings in New Brunswick and Nova Scotia. In PEI, only a small number of ticks were collected, but this is one of the first findings of ticks collected by active surveillance, which is meant to be a stronger method than passive surveillance for determining if a tick population is actually established. And so this is a pretty interesting finding that we're actually collecting them from the field. And that also considered in light with our findings that we got quite a few submissions and we are detecting pathogens, means that PEI needs to be a place that we're paying attention to and thinking about emergence of ticks and tick-borne diseases. Okay, so how do we take all of this into account? We know that a one-size-fits-all approach to ticks and tick-borne disease in Canada will not work. We have a huge geographic area, we have variations in tick populations, we have different climates, habitats, and so it can be very challenging for there to be any kind of one-size-fits-all. And that even applies on many provincial levels. Even in Ontario, risks differ very much by region, and we need to consider that. And so if you're considering developing or refining your approach to ticks and tick-borne disease, there's, four main considerations that I really think about. What species are in your area? Because that determines sort of both the level of infestation. Some species are, form high infestations and are burdens just because they're pests, and others are risks because they carry pathogens, and some are both. Distribution, we need to think about not only on a regional but local level, and that takes into consideration tick habitat and where ticks are generally found to be feeding. Seasonality, again, is important. Like I said, we saw, out west, spring being the predominant risk time, and then in central and eastern Canada, having that spring-fall period, but there's still risk in between those time periods. And so it really depends on your level of risk aversion and the patterns that you're seeing locally. So like even right now, where I am, we still have a lot of snow on the ground, but we've had a few warm days where, in places where there might not be as much snow, some of those black-legged ticks might start creeping out and looking for a blood meal after a long winter of not feeding. And so that sort of all plays into account. We know when the peak activity times are for high risk, but the other times don't necessarily mean no risk. And then lifestyle of the animal is of course important, and that takes into consideration what type of habitat they're going into, and therefore what type of tick species they might be exposed to. And so in addition to thinking about everything that you've come up with or you've seen in this presentation, there are many helpful resources for keeping up to date on tick distribution and tick risks. And so I don't mean up to date like you need to check it every day or every week, but definitely at the start of tick season, maybe at the end as surveillance data is coming in and you can see things are changing, is a really great idea. And so the Canadian Pet Tick Survey, all of our results are going to be updated and uploaded into petsandticks.com. There's a specific tab for that on the website. etick.ca is a great resource. It's currently not available in every province, but it has been expanding and it's almost national. And this is a free tool for you to use where you can take a picture of a tick, upload it, and within 24 to 48 hours, you're going to get an ID on that tick. And then there's also information on that website. All of those tick IDs end up going on a map for where they were acquired. And so it gives you a nice indication of current tick activity in a region. Of course, again, it still has all the limitations I talked about with passive surveillance being, only people who are interested are going to be submitting. It's going to be based on population, but it's still a helpful tool. The Canadian Lyme Sentinel Network will continue to publish its findings, most likely through the Canadian, the CCDR, and so openly accessible online. There is the Canadian Parasitology Expert Panel Guidelines, which are always helpful. It's accessible at this link, and you can just Google the Canadian Parasitology Expert Panel to find their guidelines. And then your provincial and regional public health organizations, particularly if we're thinking about black-legged ticks or Western black-legged ticks. These organizations are inherently worried about them for the human health risk, and so they will publish surveillance findings. What happens in each region and how that is shared is very regionally specific. Okay, so I put together a little algorithm. It's probably nothing particularly new for what a lot of you are doing in practice already. I think there's just a lot of concern, particularly in areas where they're getting more and more ticks, and you know, what do you do every time an animal comes in with a tick? And so we really advocate for tick ID. It's sort of your first line in risk assessment to know, okay, is this a tick we need to worry about or not? You can identify it in-house. So on our website, petsandticks.com, we have a video and some ID resources. There's also other training programs that you may have participated in that have been offered to the veterinary community. Or you could submit it for identification to etick.ca. If you're in one of the provinces where it runs, which is more common than not, that's a great resource for you. Or you could do both and have etick do some of that ID for you and help you develop your identification skills for in-house. Once that tick's been identified, there's a few key things to do. You know, you might want to monitor for clinical signs, and you're maybe considering future testing depending on what type of tick it is and the risks in your region. If we look at the consensus statement on Lyme disease for dogs and cats, they recommend annual testing to look at exposure. You know, every time a tick is found on an animal, that can be really cost prohibitive. And so you might want to have certain check-in points if an animal does have a lot of ticks on them. And that's a great time to talk about tick prevention and thinking about being on appropriate tick preventative. There's lots of great products out there that are targeted to certain tick species. So just make sure that the tick product that you're using is really good for the tick species that are present in your area. Advocating for tick checks. So a lot of the tick preventatives we use, they kill as they feed. So it's always great if we can get ticks off earlier. Obviously, tick prevention is sort of our front line, but tick checks are an important habit to get into on ourselves and on our pets. And then we can talk about avoidance of tick habitat as well. Of course, if you have a dog and I'm guilty of this, that just loves to go running through the woods and it's, you know, the best part of their life. Obviously, we don't want to take that away from them, but then considering tick prevention and tick checks and really putting that into priority is important. You'll notice here that I didn't mention anything about testing the tick, and this is the same recommendation in human medicine, that individual tick testing should not be used to guide clinical diagnosis. There's a lot of things that happen between a tick bite. We don't know if the animal is going to become clinically ill, if that pathogen was transmitted. It depends on tick time. If you got all the ticks, and it sort of creates a false sense of security or a false sense of worry. And it shouldn't ever be used to guide clinical treatment. We don't have any evidence to support this. And so if you're curious about some of those practices, we did recently publish, this is open access through the Journal of Veterinary Internal Medicine, that looked at some of the practices that are common among Canadian veterinarians with Lyme disease, mostly because, you know, as a veterinary profession, you know, we've been faced with so many changing guidelines. We need to know kind of what are the practices that are out there? Where do we need to focus more research on? And people still have questions. And so there's a little discussion in here about approaches to tick ID and tick testing and what we currently know. So if you're curious about more of that, I would encourage you to look at this article. And as I wrap up, and hopefully we have a couple minutes for questions, I just wanted to let you know about a new initiative that is coming your way really soon. We were fortunate enough to receive funding from Merck, Boehringer, and Zoetis to develop these infographics from the Canadian Pet Tick Survey. And so they're specific based on regions, the regions that I just provided. So some are at the provincial level and some are on the regional level, and it summarizes the key findings from the Canadian Pet Tick Survey. And so these infographics, there's going to be a subset that are printed and available through some of the representatives in your clinic, and they'll also be accessible online. And so if you subscribe to our website, now I sound like an advertisement, but through petsandticks.com, any of that information is going to be shared there. And so you've heard enough from me. If you have any questions, we can open up the floor to questions now. And thank you for being here and being part of National Tick Awareness Month.
Thank you so much, Dr. Clow. That was really interesting information, and I love the ability to take that regional approach. I think it's really going to help. Dr. Clow was kind enough to share this information with us at the CVMA and Merck to update our ticktalkcanada.com website. So a lot of the percentages that she showed will be up there as well on the interactive geography map. So we do have about 5 minutes, and we did get a lot of questions. So I'll try and pick some of the ones that I think will have the most national view. So one question was, as you were looking for your tick-borne pathogens, did you look for any other diseases, for example, Powassan virus?
Great question. So right now, our tick testing focused on the two that we're concerned about in companion animals, so that being Borrelia burgdorferi and Anaplasma phagocytophilum. And predominantly because our funding for the project, really comes from a companion animal focus. So Powassan isn't known to impact our companion animal populations, but that being said, it is a concern for human health. The great thing about this study is we now have DNA in a freezer from all of these ticks. And so as funding becomes available, we're certainly really interested in opening up some of that tick testing. And Powassan virus would be one of those key areas we'd like to look at. Powassan virus is really, it's a frustrating thing to study, or it's interesting, but maybe frustrating, because it just sort of pops up in these random places. And so having more things to test, more things to understand ecology and potentially geographic distribution is exciting. So I really hope that in the future we're able to do that.
Perfect, thank you. So the next question is around these invasive species you were talking about, like the Asian longhorn tick. Do you ever think there'll be a time when the CFIA is going to request imported dogs or imported pets to need tick prevention in order to try and control some of these invasive species coming to our country?
Yeah, that's a really great question, and there's been, I'll put on a different research hat, a lot of conversations. Dr. Scott Weiss has been a huge advocate for, different protocols for things coming in for imported dogs. And I've started doing some work with him over the past year as well, in collaboration with the CVMA and the Public Health Agency of Canada. And the CFIA has been brought into those conversations too. And so it's an active area of research and an active area of conversation. And honestly, we have a lot of data to support that we're seeing these ticks come in. It's not just a one-off. It's a pretty consistent pattern. I can't speak to whether those changes will happen, when they will happen, but it's part of the conversation, and it's certainly something that we're looking at. And I do hope that we can make some positive steps forward. If I'm going to put out a plug, we are conducting a survey shortly. It'll probably go out within the next month through the CVMA that is looking to get information from Canadian veterinarians on their experiences with canine importation and what sort of risks they're seeing. And so if you get that survey and you're here, if you could answer it, that's just going to add more to the information database and sort of the rationale for assessing what changes need to be made.
Perfect, thank you. So a question about anaplasma. You'd mentioned that in Manitoba, the percentage is higher than in other parts of the country. Why might that be? Why do we see that variability if it's the same tick?
Yeah, I don't have a good answer for that. We don't know a huge amount about sort of the pathogen invasion process, as I'll call it. There's a few theories out there that are particularly interesting. So tick first, ticks arrive, they don't have any pathogens in them, they set up shop, and somehow the pathogen gets introduced later on, if a reservoir species comes in. Pathogen first, meaning that it's already sort of there and circulating in other tick populations, or they come together. So we have ticks introduced on a competent wildlife species, and they're already carrying that pathogen. We don't know a lot about what actually happens with Borrelia, and we even know less about Anaplasma. And then there's sort of inherent things about the pathogen, about how well it survives within a tick, how well and how long it survives in a host, and therefore that impacts sort of the transmission cycle and how efficient that transmission cycle can form. And so those are questions that we have. We've started to see things, as I mentioned, pop up in Eastern Ontario where, so I'm based in Ontario and I've done a lot of field surveillance and field research in Ontario. And that's been something that over the course of that time we've noticed. And so that's an area that we want to start doing more research in to understand why that's actually happening. We know that it's slower. It seems to be slower and less efficient than Borrelia. But when those changes start to happen, we don't really know what's occurring. So right now we can tell you where things are changing, but we can't really tell you why, unfortunately.
That's all right. Well, thank you so much. We're out of time. I know there's more questions on there, and I thank you all for your participation. Dr. Clow, thank you so much for taking the time to share this very valuable information with us today. And on behalf of the CVMA and Merck Animal Health, I want to wish you all a good National Tick Awareness Month and have a great rest of your day.